And those specific antibodies, because they're so specific, if you take a patient sample, I don't know, pus, semen, blood, whatever it is, right? And you flow it over the top of it, who will get captured? Only the specific pathogen. You see that? Now, if you have a million of them, you're going to capture whatever pathogens are around, right? And so we use capture a lot. But what we like to do is we like to wash a lot. Rinsing is important because antibodies, right, have to bind. And if you rinse them, if they haven't bonded, they're gone, right? And so rinsing is important. But if you rinse and they stay, that means you've got a match, yeah? So rinsing is very important. Every technique I've ever done in lab is always rinsing. Okay, do we have to just look for the pathogen? No, we can look for what? The antibody to that specific pathogen. And one way you can do that is, again, line a slide or a tube with what? Lab-grown microbes, antigens. And then what do we do? Now we pour over the patient sample. In this case, it's going to be their anti-serum. Remember serum containing antibodies? Pour it over, and if we trap, we capture that antibody, we're going to be good. We know that you have antibodies to that disease. Because when you rinse it, if it hasn't bonded, it goes away, right? Okay. Now I want you to notice, can you see I've got an antibody to an antibody? We're smart. We're microbiologists. We can make antibodies that are specific to human. In other words, they're anti-human. Remember how we talked about blood types? And if you're type A, you're not going to make anti-A. So what does anti-A mean? It means antibodies against that A. Remember rhesus? If you're rhesus negative, you may be capable of making anti-rhesus. What's anti-rhesus? Antibodies specific to that rhesus marker. Are we all clear? So if I talk about anti-human, I'm not talking about hating humans. This is microbiology. I'm talking about antibodies that are specific to human antibodies, to the step. Okay, are we all clear on that? Okay, what can I do? Well, I can use a horse. Remember horses? Big? I can take horse antibodies and make them specific to human. It's just called vaccination, right? I can make anti-horse in a kangaroo. I wouldn't. I could make them in a dog. I could do anti-cat, anti-dog, anti-monkey, anti-cow, anti-human. And you know what this reminds me of? You know cheerleading? Look at the high school, at the middle school. You know the cheerleading, you know how everyone goes on top of, that reminds me of antibodies binding to antibodies, right? And you get a complex, don't you? All right, so can you see how this might work? Just in general, this is like my general little introduction. All right, so how do we know that an antibody has bonded? Because we're smart and we modify them in the lab. So one way to modify an antibody is to add a really nice fluorescent pigment. Can you see my fluorescent pigment there? And if this antibody binds, what comes along with it? The pigment, right? And the cool thing about fluorescent pigments is that, and we call it immunofluorescent. Can you see the immuno part? Can you see the fluorescent dye part? We call it IF diagnosis, immunofluorescent diagnosis. Because even if you have tiny particle pathogens like viruses, if you have enough of these binding, you're going to have a giant blob of what? Fluorescent dye. And the cool thing about this is we call it black light. Black light is UV. And if you put a slide, okay, with a patient sample and antibodies, and if they're fluorescent antibodies, if they bind, you'll know. Because what will happen is the whole slide, when you shine light on it, not ordinary light, remember, black light, UV, the whole slide will light up like strings of Christmas tree lights. And if the patient is negative, and remember we rinse, remember I said rinsing is important, those antibodies that are carrying the dye will get rinsed off and they won't stick around. And when you shine black light onto black, guess what?
Nothing.
If there's nothing there, it's black. So can we get a result within minutes? Yes, and that's why we love serological testing. Of course, the reality is it takes like a day because you've got to get it to the lab, to LabCorp or LabQuest or Medi, whatever lab you're using. Quick lab, there's all these good ones. So that's one form. Another one is where you add an enzyme. And I'm just going to write enzyme. So you know an enzyme has an active site, right? So the same thing. If this antibody, okay, finds the, it doesn't have to be a sandwich like that. If it finds, there we go. If it finds an antigen of any kind, and it has what? An enzyme on it, you won't know at first. You won't know at first. Why? Because can you see enzymes with the naked eye?
No.
But enzymes do what? They're catalysts, aren't they? So it's so clever because what you do is you add the substrate afterwards. So first of all, you let what? Antibodies look for the antigen, give it time, right? So you have what? Trillion of these. And then what you do is you add, what's this? This is the substrate, which is some chemical or other. Who knows what it is, right? The substrate. And if the enzyme is there and it didn't get rinsed off with the antibody, then what happens is we have designed the enzyme substrate complex so that when the substrate turns into the product, what happens to the whole thing? It changes color. It changes color. Isn't that clever? If the enzyme is there, you don't see anything. If it's rinsed off, you don't see anything. Clear. But if it binds, and then next step you add the little substrate, the whole little test tube goes a color. And you can decide, they have different enzyme substrate systems. Some go red, some go blue. I think the one in my slide is yellow, you can see. Okay? So it's real cool. How long does it take? Minutes. You just need the sample and off you go. So what do we call that? Well, what's linked to the antibody? The enzyme, right? So it's called enzyme linked. And then you see the word immuno. And then they say sorbent assay. Assay just means test. Sorbent means everything binds together. Immunosorbent. In other words, ELISA. E-L-I-S-A. Have you written ELISA down on your disease cards?
Yes.
Woohoo! Now we're going to do the final piece of the puzzle. How does it all link together? So we've got the whole course coming together, all the disease cards, all the final piece. Okay? And how do we solve, not the crime, how do we solve the infection? Okay, another way we can tell. Do you remember complement? We love complement. When an antibody binds and finds, remember, the antigen. Okay, great. We have a match, right? We have a match. So now we add some little friends of ours. And one of them will now bind to the knees. Remember our little friends? These are called, remember, complement proteins. And once you get one binding, we call it fixed. We say complement. Remember how to spell that? C-O-M-P-L-E-M-E-N-T. Complement is fixed. What does that mean? It means it binds. Why does it bind? Because, remember there's the binding spot on the stem that particularly IgGs, IgMs have, you know, different ones than others. Then what happens is that triggers the whole destructive knifing business that complement does so well. We call it a complement cascade, do you remember? And it's like dominoes. you push it, then it starts the whole cascade effect. You know, it starts to split, and you go from, and that one is usually C1, and then C1 splits, and you go all the way down to what? C6, C7, C8, C9, and then you get the mother of all. By the time it's finished, you get these, remember? What's that called?
Epitopes?
What's that called? It's called a Mac attack. Have you forgotten? What happens to that pathogen? Or to that human cell that we've triggered?
Lysis?
Lysis, right? Okay, so can you see we've got two different kind of antibody type of tests. Okay, we've got, what is it, the one where we make a pigment? What's that called? Immunofluorescence. We've got the one where we link an enzyme. What's that called? Enzyme linked. And then we've got the one where we actually add complement and we can see lysis. Actually, we can't. We have to use an indicator system, but we'll get to that. Okay, so these are the basics that you need to know before I start. Any questions on that for now? Okay, now I'm going to go through them. And I think my theme of the day is syphilis. What's the causal agent?
Treponema pallidum.
Oh, you are awesome. Treponema pallidum. What would we see under the microscope? Okay, which means what? What colour? What shape? I still don't understand.
Gram negative. Pink.
What? I don't know what colour gram negative is.
Pink.
And a spiral. Thank you, ladies and gentlemen. You're left. Because you know, you're always going to see a picture or a description of a picture. You have to interpret it, don't you? So that's always good to know. Well, does it have blobs? Does it have little sticky out things? Does it have intracellular little particles? Those are all good. All right, very good. Okay, I just do that. This is where I go back and forth with the lights. I wish it was in the Here we go. So where are we? We're on what? Serological testing, everyone? Okay, all serological tests are based on what? Antibodies binding to antigens, very specifically, and we can use it to detect things. What did I say the two versions are? Direct, indirect, meaning if it's direct, we're detecting what? The pathogen, the patient antigen. If it's indirect, we're looking for antibodies. Which is easier to find? I went through that. Antigen or antibodies? Good. Which is better for patient diagnosis, though? Finding indirect. Could you have antibodies for a different reason than you being sick today?
Yes.
Then which is going to be better? There you go. You've got to have basic understanding. Okay, so, big problem. A lot of the antibodies do cross reactions. If you're a virus, you have viral proteins, right? And so you can use an antibody that binds to viral proteins. That means you're going to get a positive no matter what viral disease it is. And we want something more specific, right? So polyclonal is what you do. You know, your bone marrow, your spleen, that's what gets activated when you get what? You know, when the microbe gets in. You produce a polyclonal response. That's good, that's healthy. The more the merrier, right? But if you're doing a very specific test, we want one, and we want a good one. And we don't want any cross reaction between like poppy seeds and like drugs when we're doing drug testing. It's very dramatic. All right. So when you're selecting which one are we going to use? Because remember, every B cell, every plasma B cell shoots a different marker. Remember, they have the markers, but they also shoot those markers called antibodies. And so I always go, it looks like a box of chocolates to me. So how are we going to select the best of the best? Well, it's like when I have a box of chocolates, I eat my favourites first. Don't you? We pick the best of the best. So how did they figure out how to do it? First of all, why do we need to do it? I hope everybody understands. We don't want a false pregnancy result, do we? We don't want a false HIV or cancer result, do we? We want as accurate and as believable and valid as possible. Also, monoclonal antibodies can target specific memory T cells, which means we might be able to interfere with organ rejection. We might be able to interfere with, well, sepsis, when you're having too much of a response. We are developing drugs that are made of antibodies that will neutralize issues. But how the heck do we do it? Well, the brilliant people came up with a way to select the best of the best. Couldn't use humans, they used mice, because that's pretty much the closest little mammal that's cheap to use and available. And what they did was genius. They took an antigen, obviously gave the mouse the shot to activate the immune system. All the B cells proliferated in the little spleen of the mouse. And then, this was the genius part. Okay, so they fused the B cells, all those B cells, thousands and hundreds of thousands of them, with cancer cells. Now, usually when you hear cancer, you're like, oh, cancer. But in this case, talk about making lemonade out of lemons. Did you know cancer cells, you can grow them easy peasy in the lab? Did you know you can grow them as cell lines for like 40 years, 50 years? Did you know that if you modify a cancer cell, you can use it as a factory in the lab? This is a factory to make antibodies. No kidding. What do we call a cancerous B cell? You need to know. You see the oma, which means cancer, tumor, see, it's called myeloma. So if you think of myeloma as a disease, you know it's that B cells that are cancerous now, don't you? You know a little bit more. So what do we call it when we fuse one for one? Each fusion gives us something called a hybrid oma. Cute little name. Says exactly what it is. Each hybridoma has a different plasma cell, right? Origin. And so each one is going to shoot different clonal versions. One clone, one cell. So what you do is you test us. You have a few years of research and testing, and you can come up with the best monoclonal antibody to detect syphilis, or the best monoclonal antibody to detect the hormone of pregnancy. They are superior to polyclonals, they're more specific, and you get fewer cross reactions, and you can choose between them. Okay? So we want to be believable and reproducible. So I'm just reviewing a few terms. I know I went through them already, but it's supposed to be clear again, is it? If you're immunocompetent as opposed to compromised, that's a good thing. That means you have the capability to form memory B and T cells, and therefore antibodies. If you're like a 1-month-old baby, are you immunocompetent? And that's why it's important to vaccinate the rest of the population. Because if granny has RSV, guess what? They can kill their grandchild. So it's very important that we protect. The RSV shot is having a few little issues at the moment. My doctor told me not to get it, even though I have a little grandbaby coming in August. So RSV, they're having a few little issues with hypersensitivities. So I'll hold off for now and wear a mask when I'm around it. That's all right, we've got time. Okay, what happens when you are seronegative? That means you haven't formed. Remember, it takes time, doesn't it? Now we say the average time is what, 10 days to 14 days? There are some diseases it takes way longer. Think of HIV. It can take months or weeks before we start seeing first antibodies, because who's being attacked? The memory T cells. And that's true of certain diseases. It can take time. So what do? We test, and then we retest just to make sure if we suspect a given disease. Because we want to make sure that when we test you, if we're testing for antibodies, we want to make sure you have them, right? They're seropositive. And you want an adequate titer. So the way we detect them is typically by what they do. We can detect agglutination, toxin neutralization, and something called complement fixation lysis. So you see it's going to come up in a minute. All right, so this is a very important one that you need to know from third unit. It comes up again here, it's not diagnostic. Which is more indicative of a current infection? Circle the correct answer. What is it? IgM. You as a student, that's your job to know that. Yeah, You'll always have IgGs, they get there eventually, but that's a good indicator. Remember, you're sick today, you didn't have the illness a few years ago, right? Or how else could you have IgGs? Vaccination, thank you. So, here's a person with Lyme disease, here's a person without Lyme disease. On the bottom, can you see the weeks, week 1, 2, etc. for both? Can you see we're looking at a specific titer against causal agent of Lyme disease, everybody? Borrelia, Burgdorferi, something with a problem with spelling on that one, isn't it? Make sure you practice. And what happens over time, if you're a healthy person and you're a sicko, what should be happening to your titer? Well, every week that goes by in this longer term disease, can you see your titer goes up? But on the other hand, if you just simply have arthritis from a natural form, you just have background noise. You see the background noise and no real change. Okay, so that's a good thing. We can do those type of tests. You know, agglutinin precipitin is where you get clumping. Remember clumping? Okay, what's the difference between the two? Very, very simple. This one is liquids. Can you see to precipitate is from chemistry, where you add one liquid to another and you get like, you know, a deposit. Whereas an agglutinin means you have tiny particles which you actually clump, so solid to bigger lattices or clumps of solid. Okay? But they're the same thing and they do the same job. And so that's all that says. Okay? Very good. And who's good at doing this? Same old IgMs and IgGs. All right, so when we do a ring test, what we're doing is we're putting, and again, okay, so if we have antibodies, and I can turn this around and have them going this direction, you know, I'm just going to do Y's, right, in one area. And we have, these are the antibodies. And then we have a large well, or area, that are just antigens. You heard of something called the D word, diffusion. Okay, so if we have a liquid tube, in which case we're turning this like this, there's our test tube, you see an area with antigen. And then you see an area with antibodies. What happens? The D word?
Fusion.
You can't see it right now, right? You can't see it. Where they meet, they call it in science, they call it the zone of equivalence. Meaning, you're getting a good ratio, so everybody's finding each other, right? Because to begin with, you're not going to have many of each, but as they keep diffusing and diffusing, you'll get more and more of them are pairing up. And so what will you see in the tube? You'll see a precipitate. Can you see this is a liquid? Because what's happened? These have gone this way, these have gone this way, and where they meet, okay, it won't be red, it's typically precipitates, usually white, very boring. Okay? You can do the same thing, okay, in a, same thing as that, but in an agar plate. Do you remember the Kirby-Bauer test? Remember the Kirby-Bauer test? In this case, we can put, for example, antibodies in the middle. Okay, so that's these guys, right? And then we can put a clump of antigens around the outside, right? But the same thing happens. What happens? These guys, you can't see them in the agar, but will they do the D word? Will they diffuse? Then these guys will be going out like this, right? Like diffusion. These guys will be going out like this. And then where they meet, we call it, remember, it's called a zone of equivalency. There'll be lattices and clumps that we can see. And what will it look like? Same thing. Like that. Because wherever they meet, you get, what have you got? Everybody understand? Okay, zone of equivalence. So check it out. This one is liquids. Can you see, it doesn't matter which end you have which. In this case, can you see there's antibodies on the bottom? You can't see it. Can you see there's antigens on the top? You can't see it. But in the zone of equivalence, see a line. We see the line. Okay, we good? Yay! Precipitation, everybody. Immunodiffusion. Oh my gosh, now I've turned it around. You see I've got an antigen, okay, and then I've got antibodies. You can do it any way you want, but the point is, can you see where one diffuses? It's going all over the place, and the other one goes all over the place. Can you see a very faint white line there? You see the white line? And you can see on the bottom there's another little white line. You see, that's the zone of equivalence. So if we're testing different patients, serum just means a patient sample, right? When we test different samples, can you see patient number 2 is negative? Patient number 3 is negative. Oh dear, patient 4 has histoplasmosis. Because where they meet, we've got an issue. So patient 4 is the patient, and can you see patient 1 is not a patient but a control. Don't you always need a control? You want to make sure it's all working properly. Because antibodies are proteins and they denature, so you have to make sure the system's working. Okay? One that we do on a slide specifically for, do you remember Streptococcus pneumoniae? What disease does it cause? It causes pneumococcal pneumonia. It's AL, okay? Very close, well done. Pneumococcal pneumonia and missed the second disease. It's on my disease sheet. You do not know your name. Shame on you. You're going to fail that quiz unless you go through it 10 times. Yeah, but that's not the one that's on my disease sheet that you have to know. Otitis media, you noonas. Completely different. Nothing to do with the lungs. Nothing to do with the brain. Oh my God. You've got to know. What's it to do with? What's otitis media to do with?
Middle ear.
As a different organ. It's called the sensory organ. We do them at a different point in anatomy and physiology. Sorry, I'm shouting at you and I'm laughing because I'm just going, they've got time to study, so I don't feel bad about yelling at you. You've got time. I figure if I yell enough, you'll study more. Sorry. Okay, so the Quellen test is Streptococcus pneumoniae, and what we do is we put the pneumonia on a slide, and then we add antibodies that do coagulation and clumping. And the deal is, the antigen in this case is, in case you didn't know, name, picture, what does Streptococcus pneumoniae look like? What?
Coxsackie Shane.
Chains or they're often in pairs. Good. Oh, very good. What do they have? Morphology doesn't just mean wall. You have to know morphology. That means if you don't know it, you haven't done a proper drawing. What else does it have that's real obvious?
Purple.
No. It wouldn't be purple. The wall, I said besides the wall.
Oh.
Okay, besides what? You're right, it's purple, the wall, if you
Yes.
stain it. But I'm saying if you look at it, what else do you have that's to do with the cell morphology?
Does it like plop, 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 plop?
We already got that one. Done the boop, 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 boop. Well done, we covered
Okay.
that one. Or boop, 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 boop. Good job. It's called a capsule. And if you haven't drawn your capsule, then you will fail.
Oh no.
You will fail. It's like when you do your viruses. Don't leave out the envelope. Remember, morphology means what you see. It does not just mean monovision. We're not going monoclonal here, everybody. We're going poly. You've got to know the features of what you would see where? Under a microscope. Okay? So it's structures. Are there other structures you should be thinking about? Of course. Didn't we do chapter 4? The cell. Did we do chapter 5? The eukaryotic cell. What are the features? Just the basic. What will you see? Nothing complicated.
Blob.
The blob is always good, or blobs.
Blobs.
Because there may be more than one blob, and that's a giveaway. But you've missed something very important. How do we classify certain groups, certain kingdoms? How do we classify an individual kingdom? I was sitting in Bio 1. They all knew.
Cell wall.
We've done that. Good, good, good. Motility. All the Bio 1 people knew that. I was in a study group when we had the lockdown because we couldn't get out of the building, of course. So we did a study group in Bio 1.
Same with us.
It was awesome. But if you know your Bio 1, even they're going through it, going, right, what are the features of cell morphology? So please don't just think it's wall, okay? Make sure you know it's what organism, what do you see? Good, all right, enough nagging. All right, it's a capsule, and Streptococcus pneumoniae cannot do the A word without it. In fact, we had a whole experiment that was all about, it was called Griffiths and Avery, and it was all about the capsule. Oh my gosh, Streptococcus pneumoniae. Oh my gosh, it's back again. You'll hear it several times. Anyway, they swell up. If the antibodies bind, the bacterium swells up. If it's not strep pneumoniae, it doesn't. It's called the Quellon test, and it's on your disease card. All right. Latex agglutination, you've seen it. Remember, if it's there, it clumps. If it's not there, it doesn't. And we enlarge it because we use latex that's coated with antibodies. And that's how we know. Not used quite as much as usual, but I hope you can tell which patient is clumping. You see, you can tell by the clumping, everybody else is negative, and you can figure out that. Usually we use IgMs, IgGs. All right. Do you know there are some nasty viruses that clump? There are some nasty viruses that clump the red blood cells. We call it what? Hemagglutination. There's some real nasty viruses. One test is to see if we can block that reaction. So we can actually put it on a slide or in a well and see if we can block the clumping. If we block the virus, then it won't be able to hit the red blood cells. So can you see the virus is there? Can you see it's clumped the red blood cells? But if we use antibodies, there isn't a picture of it, sorry, I would draw it. If we have antibodies that bind and neutralize the viruses, can they clump the red blood cells anymore?
No.
And so again, you can tell by using an assay like that, stopping the clumping. All right, so this is where we want to talk about complement fixation. It's called the classical. Remember when an antibody binds to an antigen, then it triggers the classic pathway. Who binds? Can you see C1? And then we go all the way to the complexes, and we know that you get more inflammation in a body, but we're in a test tube now, everybody. We're in a test tube. And so, let's talk about somebody who might have syphilis, and somebody who hopefully does not have syphilis. And so when we look at we can do a direct or an indirect test, but syphilis can be quite hard to find. So we quite often will do the indirect just for this particular pathogen, but you could go either way, okay? So let me go ahead, I'm just gonna write at the top, we've got a patient that we think is positive for syphilis. Potentially. Remember we do a presumptive first, and then we do a test. And then we have a patient that we think is negative. Okay? So, what's the first thing we do? We collect a serum sample. So I'm going to put the light on so that we can go step by step. Now, when we do a complement fixation test, if you have the pathogen, if you have the antibodies, then you'll get a match, right? Whatever system you're doing, antibody to antigen or antigen to antibody, if they bind, you've got a match. Because do you use specific antibodies or use specific antigen when you're doing the test? Right, very good. So when you do the specific test, what will happen is if you allow complement to work, what will it do? Well, it'll burst the syphilis organism, won't it? The trouble is, if you do it in a test tube, it's invisible. You can't see it. You can't see it, and that's the problem. But we can use this test if we put a second indicator set of cells in the tube. That second set of cells is usually red blood cells. Okay, let's talk about blood for a minute. Okay, let's review blood. If I put some blood in a test tube, can you typically see through it? Okay, it's cloudy. It's red and it's cloudy, right? But if I lyse it, if I add anti-RBCs, you know, RBC means what? Red blood cells. If I add anti-RBCs, remember what that means? That means antibodies against red blood cells. If you put the two together, what will happen? They'll match, right? They'll match, they'll bind. And if they bind, and I add complement, what will happen to those red blood cells?
Lice.
They'll lyse. Do you know what lyse blood looks like? It looks like peptinization. Do you remember that? So literally, when you hold the tube of blood up, it's still red, but instead of looking, cloudy, it looks like blood juice. Okay? Can I just call it that? Blood juice. All right?
Yeah.
And so we use it as an indicator system. And we don't use human blood, it's too expensive, you know how we go. Instead we use sheep blood, which is very available in the United States, and it's cheap, okay? And we've got antibodies. Remember, you can form antibodies to anything, but you can definitely form them to sheep red blood cells. Okay, so I've got you primed, okay? Right, so as I said, I could teach this to you really, really fast, but it takes time to explain it properly. So let's start off with the first step. It's just a bummer that these lights are out. I'm so sorry, everybody. Stupid old RCC. It's like it takes 6 months to order the system. You know the old-fashioned light bulbs you could do the same day? Well, because we're a multi-million-dollar building, it takes months to get the right thing, and then they have to pull everything down, and it's a whole. Anyway, that's another story. I don't mean to sound bitter and twisted on that one, but anyway. All right. Yeah. So anyway, best than not having any lots of quite right. Good way of looking at it. Lovely. All right. So let's say we've got positive. So what we do, the first thing we do is we collect the patient anti-serum. Does everybody know what anti-serum is? That's the blood. We take a blood sample. And we don't want what? The blood cells from you, the human ones. And so it's just the anti-serum, and it doesn't have the normal, what you have in your blood. Remember, you have complement. So it's without complement. And that's a very important thing. So we collect the patient anti-serum that does not have any complement in it. So the only thing we're looking for is what? Antibodies, because this is going to be an example of an indirect test for you. Okay, so this patient we think has syphilis, and indeed he or she does. And so when we look, we see trillions of antibodies, but you can't see anything because where are we? We're in a little test tube, yeah? We're in a test tube, you can't see anything. So do you have antibodies if you're positive? You should, shouldn't you? If you are negative for syphilis, you should not have any of those IgMs or IgGs, whatever they are, at that point. So no, right? Everyone okay with that? Okay, that's step 1. Okay, now step 2, we add, I think I'm just going to stick this one. Step 2 is we add the antigen. So we add the antigen, and let me remind you what the antigen is. Well, we're looking for, in this case, the syphilis organism. So the antigen is lab-grown syphilis. Are we all clear with that? Lab syphilis. And so what am I going to draw, everybody? Well, we're not staining anything, so don't tell me pink, okay? But what I am definitely going to see, right? And we do it to all our patient samples. We've only got 2 patients we're testing, but normally you'd have maybe 500, whatever it is, okay? You next add, what did we add? We added the antigen, the syphilis organism in this case, right? So now what we do is we give it time. Da-da-da-da-da-da. And what's going to happen? Well, these guys are going to meet and bond. Isn't that the whole point? There's a match. So we'll have this. We'll have this. And we'll have this. What do we call this? We call it an in any order you want. Antigen, antibody complex, right? Meanwhile, what will we have here? Same old, right? Are we all clear on that one? Awesome. So now, what we add is we add complement. When I say complement, that's like millions and millions of those complement proteins. Is everyone clear what I mean by complement?
Yes.
So what are we adding? And remember, it's all in here, right? And a different test tube over here. So what are we adding? Now what's going to happen is that the complement, you've seen this one, will be, what's the word that means it binds?
Agglutination.
That's not it. It's complement is what? What's the name of the test?
Complement fixation.
So the verb?
Fixation.
Thank you. No, the verb is not fixation. You're
Fixing.
It is fixed. Watch that on your T's test. That's what they're looking for. Warning you. Okay, so what happens? Let me just draw it. Because now what happens? You've added complement, and what does it go for? Well, you've got all these little guys, right? Here we go. Like this. And where does the complement go? Remember the knees? It goes there, it goes there, it goes there, and then everything goes berserk. And what happens? Well, we say complement is, let's make sure we get the verb right this time, is fixed, which means that what's the end result? What's the end result? If the complement is fixed, what happens next? Mac attack. So what's going to get destroyed? Are we all clear? What are we going to say on this side? Not going to solve it like that. And I'm going to say not fixed yet, because we're going to give it an opportunity. The problem is we can't see this versus this. Okay? So we have to do a 4th step that's called the indicator step. You know how indicators help? You know, like when you add reagents? So now we have to add a special reagent. We have to add the indicator. And the indicator is the system of a second cell anti-cell set. So what are we going to add? We're going to add RBCs, you know what they are, red blood cells. And we're going to add anti-red blood cells. What does anti mean? Antibodies specific to red blood cells. Okay. So what are we adding? Can you draw a red blood cell? Right? And then we're adding what? what's going to happen?
Back.
Okay. All of this is all of my PowerPoint slides. Every step. By the way. Right then, so let's consider what's going to happen. First of all, by this point, we've got a test tube that's literally, what, cloudy with blood in it. Everyone know that's what we're starting with, so we add that in. Now what's going to happen? Well, in this case, all the complement has been, what's that word again? What's the verb? Fixed. Okay, so is it available? Does it form, remember the Mac attack? So what's going to happen to this blood? Nothing. Why? Because complement has been used up, it's been fixed, right? So can it destroy the red blood cells? Leave it alone. What about here though? Do we have complement available?
Yes.
Not fixed, is it available?
Yes.
Okay, so what's going to happen? And then, okay, ready? Okay, so what are we physically going to see in the test tube? Well, can you see in this case, the pathogen was MAC attack, but the indicator system was left alone. What does blood look like when it's left alone, everybody?
Cloudy.
Red, and I'm just going to draw it, red and cloudy. In this case, there was no pathogen to destroy, no MAC attack, because you have to have both the antigen and the antibody. In our indicator system, we have antigen-antibody, and luckily for us, blood that's been lysed is easy to tell, because when you look in the test tube, what you'll see is that translucence. Can you see the lysis? Because who's lysed? The one where we've got both present. You've got to have both present. So if we see cloudy, then is the patient positive? Yes. If the blood is lysed, not the pathogen, then the patient is negative. Right? So, very good. So here we go, complement fixation test. Test is based on the lysis of an indicator red blood cell system. Do you see where that comes into play now? Okay, lysis, right? Okay, yeah, lysis. Okay, these are the steps. I promised you. You can do either a direct test or an indirect test. It doesn't matter. I just chose to show you the indirect test. What do we add first? The serum. Then we add the antigen. Then we add the complement. Then we add the indicator system. The deal is, you observe the tube of blood. If the RBCs escape being lysed, that means the pathogen was lysed, then complement was fixed, and you can tell. If it's negative, if you're negative for the disease, the RBCs are lysed, and so on. So I put a picture there. Can you explain it? I prefer doing it on the whiteboard. Oops. So here we go. The real world. The tubes on the left, the 3 patients on the left, do they have syphilis? Are they positive or are they negative for syphilis?
Positive.
So the ones on the left are positive for syphilis, and the ones on the right are negative for syphilis. Do you understand why?
Yes.
Yes. Yeah. See? That's the one, if I were to spend 2 minutes going through it, you'd be like, well. Okay. Complement fixation is still used for certain diseases. We use it less and less now because we've got better tests. Okay, remember I talked about direct versus indirect? Remember I told you that we can make antibodies to antibodies? Remember that we can have an antibody to capture, an antigen gets captured, and then we can put, remember the modified one that has the label on it. That reminds me of the cheerleading thing. thing. This is in a test tube. So we can, there's lots of tricks of the trade everywhere. So here's the example of syphilis. Remember I just asked our little disease agent of the day. We get a sample, and then what do? We add the antibodies that have been modified. They bind, you rinse. It makes it all light up. You see the light up? If you rinse and they all fall off, they didn't bind, you've got a negative patient. Real easy to tell. We use it for gonorrhea, yay, chlamydia. Yeah, if you get a chlamydia test, yeah, and so on. All right, we can do the indirect method. Again, what do we provide? We provide the antigen. What are we looking for you? We're looking for antibodies in your serum. You bind, and then we have to add another antibody that's the labeled one. You see again? Modified, now it binds to the human antibodies and it lights up. Here's an example of syphilis again, using an indirect method. So yeah, we can do, it depends on the pathogen. COVID-19, we did an indirect test. Can you see the indirect test? Where we took a sample and we can actually literally see what's happening. So you may or may not have had that. Here's an example of patients on a smear, and can you see it lit up? So that patient is positive for antibodies. Okay, this was an indirect test as well. Yeah, same principle. Okay, can we use an antibody labeled with an enzyme? Yes, we can. And we can tell, can you see my control in the top left and bottom left hand wells? Can you see they went yellow? They are positive control. Can you see my negative control next door? My negative control next door is clear. And then can you see all the people who have HIV? Okay, this is a block where one well is one person. So we can do a whole bunch very quickly. And again, we know because it binds and we get a positive reaction. Remember that it changes colour. If we're talking about HIV, if we're testing for AIDS, no point looking for the virus, you can't find it. We have to do indirect in that case. Okay. And we'll get a coloured product. We also use it for some of the hepatitis. ELISA is very, very commonly used if we have a system. And can you see there, can you see where it's clear? We can't actually see the antibody-antigen reaction, but as soon as we add the antibody that's got the enzyme, and now we add the substrate, can you see we've got the pin change? So it's a series of steps. This is a direct test, where we captured, that was detecting measles, that's the measles virus. So you know. And again, can we do it directly or indirectly? We sure can. Indirect. Okay. When I take a sample from you, you know that proteins are really important. Antibodies are made of protein. Remember we call them immunoglobulins, like globular proteins. Did you know that if we use, for example, chromatography, or if we use a modified chromatography method called electrophoresis, something that you probably did in Bio 1. If you didn't do it, you obviously took Bio 1 during COVID, bad luck. But it's really lovely to see in electrophoresis. And what you're doing with electrophoresis is you are separating molecules by size, charge, and shape. Okay, so we separate by, what was it? Size, charge, and shape. Now, if we do normal diffusion, it's slow. You know how slow it can be? So what we use is we use an electrical current because it makes it happen in real time. We can actually push everybody. It's like me nagging you. It like pushes everybody forward. So let's just talk about that for a minute. Say this is the start line. And we have a protein is loaded onto our little chromatography paper or jelly or whatever it is we're using, okay? If I'm a big molecule and I give us 6 hours for me to, diffuse, but we zap me in the butt, so off I go, right? If I'm a big molecule, at the end of 6 hours, I may only get to here. Because big, slow. Remember we talked about that with diffusion. Even with electrical impetus, it's slow. If I'm a tiny molecule the size of my nose, in those same 6 hours, I get all the way to the back of the room, equivalent, within the same time frame. So again, big, slower, small, faster. So molecules have charges. You know proteins can have amino acids, that are, you know, have different characteristics and different polarities. If we're doing DNA, you know, each nucleotide in the DNA has a phosphate negative polarity. And so you can really shift DNA. And again, big pieces would go what? Not so far from the start line. Tiny pieces go a heck of a lot further. All right, so we're talking immuno. Immuno, we're to do with antibodies made of protein. And we're using electricity. And can everybody see? Here's the start line. Can you see the start line? And can you see we zap it this way? So can you see this would be a larger protein? Smaller, 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 and of course other things like polarity. We can then stop, and they have something called pulse electrophoresis, which means that we turn our paper or our jelly around and we zap it this way now. So what happens, each of these blobs, right, of a specific group of sizes now gets separated further, further, and further. And so you can, and they'll even turn it a third time and then zap it again for half an hour. And that allows us to separate very similar sized particles. And it's a very cool way to, you know, separate things out.
Can you do that with your DNA, like DNA amplification?
You absolutely can do that with DNA. You can do it with that. It's chemistry. And so long as the chemical has polarity or a charge, we can move it with electricity to the poles. So we're looking at anti-serum. Remember that contains the antibodies, so you can separate out your different kinds of antibodies that way. Antibodies are proteins. And so again, size, shape, and charge. Everything I just said, yeah? And what do you see? Well, did you know we can stain protein? You know there's a stain, like a reagent we can add, just like you did in your lab. And it will show up, because normally when you look at jelly, it all looks the same. But if you stain it, you can see the different blobs of proteins. We don't call them blobs, everyone. We call them bands.
Bands.
Yes. And we get, you know, different bands. So there's something called the Western blot. Okay, so what we've got here is we've got a person who does not have AIDS at first, and then can you see after 9 days, 14, all the way up to 30 days, we can test for the proteins, and we can zap. Can you see the start line is at the top, everybody? The start line is at the top. And so what we're going to find is the bigger proteins, can you see the bigger proteins, are going to be closer to the start line. Can you see 160? And then can you see we're going all the way down to 17? See, that's the smaller, shorter. And this is a control. It has every size imaginable. Bands. And we call them bands. So we're looking very specifically for the antibodies for HIV. Okay, and we can do that very specifically. Can you see at the start line, there ain't nothing there. Okay, that's just normal serum protein that everybody has. But can you see after 9 days, and then 14, can you see more and more bands appear? And can you see over time, can you see how the bands start getting thicker and thicker? That means there's more, right? There's more protein there are, the more that's stained, you get a bigger area. It's as simple as that. Now, as you know, when we do for HIV, we typically do ELISA first. And one of the issues is we may well get false negatives. Because what happens? Well, it's attacking your helper T cells that help you to make what? Antibodies, right? And so ELISA, that first one, is not as sensitive as this one. This one is a more sensitive test. And so often they'll do a follow-up with a Western blot, and that can really confirm what's going on. But even then, if you're severe AIDS, you may not, you might get a false negative now and again. But nevertheless, we do the cheaper method first, and then we follow up.
And for this one, you have a basic comparison, right? To compare the size of someone that has HIV.
This is the comparison. You know, everybody in the whole world in this field knows which bands should appear. Each country selects different ones, but in our case, we're looking at, let me see, separation, and we'll get bands, very specific, and you get that follow-up test. And we're looking for, in this country, can you see, we look for the glycoprotein 160 or 120, can you see? Or P31, but different countries have slightly different detection methods, but they're all the same kind of.
Yeah, same process.
Same process. This one is in vivo, meaning we test your skin, your body, and this one I'm not going to talk about because I've covered it already. This is the MAN2 test, remember the protein? Yeah, and we do the induration. Instead, I want to finish off with the most expensive test, the DNA, RNA test, the genetic test, and we can even do mitochondrial analysis. We can do all sorts of things. I want to start by just reminding you that we can do an electrical separation method of different sizes of DNA now, right?
Yeah.
And so when we do different sizes of DNA, it's just simply called DNA electrophoresis. So does everybody see where the start line is?
Yeah.
Different can you see there's the control in this case, and then can you see each patient we see different bands. Remember it's called banding patterns? And depending on your status, you will have different banding patterns of DNA, because we're looking for genetic material to be present this time. We can do even, who's your mama, who's your dada? Well, we know who your mama is, but
We need to find out like criminals.
who the daddy, and depending on the pattern, like they've done all sorts of things where they found, people were selling hamburger, and it turns out it contains horse meat.
Oh.
Lovely. This test. You can look for syphilis, DNA this time, okay? So very, very practical and solve crimes, but you need enough DNA to do that. So again, same thing, right? You have what, a substrate, not paper this time, it's definitely gel. It's a different kind of gel than we use with the proteins. Same thing though, a current, and we move our DNA to the opposite point because it's negative, slightly negative. Again, short pieces. Where's the shortest piece? I hope you can see it's down here in the band. And which is the longest piece? Can you see? It's right here. That was the start line of this example. Okay. So how do we know? How do we know that something has happened? Well, again, remember how I do antibodies? Me running crazy person again. Whoa! Okay. So now, what I want to talk about is DNA. Put the light on. Here we go. So DNA, this is where we go. DNA can be expressed as what? RNA. So some tests, we're more interested in which genes are being expressed. So we don't look at the DNA that's present, we look at the messenger RNA that's present, right? If you want to know which genes are being expressed. Or we might want to know what kind of